Hello friends and welcome to my channel EG Life Science. Here I am talking about flagella staining by a Bayes method. So please subscribe my channel EG Life Science, like my video and share it to your friends, and don't forget to press the bell icon so you will get the notification of newly uploaded video. So let's start. The flagella are hair-like helical appendages that come out through the cell wall and responsible for a motility of organism. Many rod-shaped bacteria and a few coccus-shaped bacteria like Planococcus and Nitrococcus possess a flagella, and by a mean of flagella they are motile. The organism which has a flagella are called as a flagellated organisms, whereas the organism that does not have a flagella are called as non-flagellated organisms. The flagella are unbranched, long thread-like structures which are composed of a protein flagellin, and they are embedded in a cell envelope. The diameter of a flagella is 10 to 30 nanometer and the length is 5 to 16 micrometer. They are responsible for a bacterial motility. Motility plays an important role in a survival and ability of certain bacteria to cause a disease. Then the types and example of a flagella. On a basis of a flagellar arrangement and their number, the flagella are of the four types: monotrichous, amphitrichous, lopotrichous, and peritrichous. The monotrichous flagella means the organism possesses a single polar flagellum, for example, Vibrio cholerae. Amphitrichous, the organism possesses a single flagellum on both the sides. Example of it, Reagents faecalis. Lopotrichous, tuft of a flagella at one or both sides. Example, spirillum. And then fourth, peritrichous, numerous flagella all over the bacterial body. Example of it is a Salmonella type. So in this image, there is a flagellar arrangement: peritrichous, monotrichous, lopotrichous, and amphitrichous. Then the parts of a flagella. Each flagellum is consisted of three distinct parts: filament, hook, and the basal body. The filament lies external to the cell. Hook is embedded in a cell envelope. Basal body is attached to a cell membrane by a ring-like structures. The functions of a flagella: for a motility or a movement, sensation, signal transduction, adhesion, and the flagella are accepted as a virulent factor. As the flagella are virulent factor, they increase the pathogenicity of the bacteria. Flagella are very thin and delicate in nature. Thus, the flagella are observed under a light microscope by using a facial staining technique. And for flagella staining, the Bayes method is used. In this Bayes method, the diameter of a flagella is increased by coating them with a mordant, that is a tannic acid, and then stained with a basic fuchsin. In a Bayes method of flagella staining, requirement 80 hours old culture of a flagellated organism, that is Pseudomonas aeruginosa, a culture tube and sterile distilled water, clean grease free slide, staining reagents, solution A, solution B, and solution C. Solution A is made up of tannic acid and ferric chloride. Solution B is made up of basic fuchsin, concentrated HCl, formalin, and ethanol. Whereas a solution C, it is also called as ZNCF, that is a Jill Nielsen carbol fuchsin, and it is made up of basic fluoxetine, alcohol, phenol, and distilled water. So these three solutions are used for a flagella stirring procedure. One slant is taken, a slant of actively growing culture of a Pseudomonas aeruginosa, and it is half filled with a water or sterile distilled water, kept at a room temperature for half an hour without a disturbance. The slant of Pseudomonas aeruginosa, after a half an hour, a drop of suspension is placed on a slide and held in an inclined way so that the suspension runs along a surface of a slide very slowly by holding a slide at a 70-degree angle. Then the slide is air dried. Then the fourth step, it is stained by following a Bailey's method. The solution A is applied on a smear and remains on a slide for 3 to 5 minutes. After 3 to 5 minutes, the solution A is removed from a slide and a slide is added with a solution B for 7 minutes. Then smear is washed under a tap water. Apply a solution C and allow it to stand for 1 minute with a gentle heat. Here a slide have to kept in a steam. After it, smear is washed with a tap water, air dry, and observe under oil immersion lens. The Bailey solution A consists of ferric chloride and tannic acid. Ferric chloride acts as a fixative and a tannic acid as a mordant. Bailey solution B consists of basic fluoxetine, ethanol, hydrochloric acid, and formalin. When tannic acid and a basic fluoxetine is heated in an alcoholic solution, alcohol evaporates and colloidal precipitate will form, which deposit on a flagella and increase the apparent diameter of a flagella. The HCl will lower the pH of the solution to the 5. The formalin serves as a preservative, and the solution C, that is a ZNCF, stains a bacterial cell. At time of observation, you can look for red colored flagella and pink colored cell. Here is the observation. Pink or a dark pink colored flagella and a red colored cell. So thank you for watching my video. Please subscribe my channel Easy Life Science, like my video and press the bell icon, share it to your friends. Thank you once again.